Imagine buying a machine with 3 billion individual parts, but it arrives in pieces. You would expect a massive instruction manual to help you put it together. Well, your body comes with one. This manual contains the directions to build and operate every single part of you. You carry a copy of it inside nearly every cell in your body. We call it DNA. The directions are written in a surprisingly simple language. Instead of a 26-character alphabet, this code uses just 4 chemical letters, A, T, C, and G. Their specific order spells out every instruction your cells need to grow, function, and repair themselves. But here is the problem. For almost all of human history, we couldn't actually see these instructions. We possessed the book, but we were completely unable to read it. DNA sequencing is the laboratory process we use to finally decode this text. It is how we transform those chemical letters into information we can actually understand. In the 1970s, researchers developed the first complex chemical methods to start pulling this code apart. This is what those early results looked like. Scientists had to look at exposed X-ray films and manually count these dark, vertical bands, one by one, to figure out the sequence of the code. Using this method was like standing in a dark library and trying to read an entire encyclopedia by shining a tiny flashlight on one single letter at a time. Researchers proved the manual could be read, but this approach was too slow and technically difficult to be used for everyday medical discoveries. Next-generation sequencing changed the speed of the game. It's the digital scanner upgrade for biology, allowing us to read the book of life millions of times faster. First, the machine takes the entire DNA manual and breaks it into thousands of tiny, manageable pieces. To make the chemical signal loud enough to hear, we make millions of identical copies, grouping them together on microscopic beads. The machine reads these pieces as they rebuild themselves. By watching the fragments grow letter by letter, the instrument can track the sequence of millions of different sections at once. Every time a new A, T, C, or G locks into its proper place, it gives off a microscopic flash of light. It is like a stadium full of people holding up colored flashcards, where a single panoramic photo captures everyone's card at the exact same instant. By moving from reading one single letter at a time to capturing millions of sequences simultaneously, we can now decode an entire human genome in a matter of days. But having all this data is only useful if we know what to look for. Why do we need to see every single letter? Out of billions of letters in your code, one tiny typo can cause disease. A mismatched letter changes cell function. Computers take millions of short fragments and overlap them until perfectly aligned. This creates a master sequence where that one hidden typo stands out. This means doctors no longer wait for a disease to advance before they understand it. They can scan a patient's code to find the exact molecular error causing the problem. Instead of just treating the symptoms, modern medicine can now target the individual root cause directly at the cellular level. We are no longer guessing at the source of our illnesses. We've gone from being unable to see our instructions to understanding them in high definition. Humanity has finally learned how to read the book of life. Now that we can clearly see the instructions, we are finally learning how to